All right, welcome everyone to another episode of 10 Minutes with JPIDS. I'm your editor-in-chief, Ravi Jhaveri, and I'm really thrilled to have two dear friends of mine who are really smart people too. Um, First is Lisa Akhtar, who is an assistant professor in our division at Lurie Children's in Chicago, and then also Anita McElroy, both MD PhDs, although I would call Lisa an MD PhD and I will call Anita a PhD MD, as I've often said to her, and they'll tell their stories and they'll explain why I say that. But we're here today to talk about a new article series that we've launched in partnership with the Journal of Infectious Diseases, talking about neurovirulence of different pathogens. And so I'm very excited about this venture, and JID has been a terrific partner for us. And so I want to start by thanking both of you for taking this on and for building our basic translational and pathogenesis repertoire for JPIDS. All right, so I'm going to start with the questions I always start with. I'm a superhero nerd, and every superhero has an origin story. So Lisa, let's start with you. So what was your ID origin story? And then the second question after that was, what was your first paper that you published?
Yeah, so I actually, before I became interested in ID, I was more interested in the brain, I think. And so I'll sort of explain how that relates to the first paper that I published. But I started my scientific career in a neuropathology lab and as a technician before I started medical school, and then got really excited about both medicine and science and ended up doing an MSTP program or an MD PhD, combined MD PhD program. And the first place that I got really excited about infectious diseases was in my PhD lab. I was focused on studying neuroimmunology, and it just so happened that the way that I started studying that was looking at the immune responses in the brain to HIV, so human immunodeficiency virus, and looking at the ways in which HIV generated an immune response and how that was thwarted by the virus. And so I really got wrapped up and excited about sort of this interplay between the virus and the host in sort of evolutionary time, and how those could compete with one another, which is obviously clearly related to this series that we've launched into now. But actually, the first paper that I published was I was a middle author on a paper from the neuropathology lab that I referenced to begin with. It was actually a paper about looking at a mouse model of an inherited prion disease, and looking at cell death, neuronal death related to prions, inherited prion disease. And I spent a long, long time staring at the brain of mice under a microscope, and measuring thicknesses of different layers of the brain, and really becoming fascinated with the brain and sort of the beauty of the brain at an immunohistochemical level.
I love that. And it makes me really think about sort of the imprinting that happens to all of us with experiences that maybe we don't even realize that so heavily influence us going forward. And I could tell you some Hep C stories about my med school and the like that I didn't really think about at the time, but ultimately, obviously, must have influenced me in some way in choosing my field. All right, Anita, so your origin story and first paper, and obviously now you've got to tell everyone why I call you PhD MD.
Well, obviously, that's part of my origin story, right? So I fell in
Of
love with
course.
virology. That's my first love. Love viruses so much. They're absolutely fascinating. And I was really inspired by Susan Dovin, who's was a professor at the University of Maryland who taught basic virology. So nothing crazy, like an undergraduate class. But she was just an amazing instructor. And her love for the subject was actually quite infectious, and it inspired me to love viruses. So I left there and decided I really wanted to get a PhD, and went out to UC San Diego, where I studied human cytomegalovirus. That was the virus I landed on for that work. And then decided to walk away from the DNA viruses, no offense, DNA viruses, because I fell in love with RNA viruses, which is still my passion to this day. And I did a postdoctoral fellowship at the U.S. Army Medical Research Institute for Infectious Diseases, looking at emerging RNA viruses, which is the type type of work that my lab still does. And then I decided, I think I want to be a doctor.
Badass RNA viruses. So yeah.
Badass, exactly. And then I decided I really think I'd be a better scientist if I was also a doctor. So I went back to school and I got my MD and did the route that we all did here with pediatrics and then pediatric infectious disease. And it was obvious to me, based upon my love for viruses, that infectious disease was such a good fit from a clinical perspective. So that's how I ended up there. My first paper, I'll talk about my first primary paper, the one I was the first author on. So this is from my grad work. This was in human cytomegalovirus because that's the work I was doing at the time. And we were studying how the virus is able to modify the host cell cycle to make the cell stuck in the G1 to S transition. And so this basically provides the virus with all the things it needs to make more of itself. So it's really a clever strategy for the virus to sort of overtake the host and, it essentially use the host to generate more of its progeny. And so that's where sort of my host pathogen interaction love began. And that also plays into some of the work that Lisa and I have been doing in this series.
Love it. Thanks for sharing that. All right, so the next question I wanted to sort of cover was actually to tell the origin story of this article series. So, Lisa, do you remember how we started this conversation? Because I do, if you don't, but it's not a test. I just want to see.
Yeah. I mean, do you want to tell how this got started, and I can kind of fill in the bits that made me excited about it, or
Yeah, let's do that. So I actually think you and I were in clinic either the same day or you had an ad hoc clinic patient and you showed up, and I was just was thinking, sort of brainstorming, which I freely associate all the time. And so I said to Lisa, I'm like, do you think people would be really interested in a neurovirulence like article series? Like, couldn't we put together something for JPIDS and you could tell your herpes story and your herpes scientific story, and then we could collect some others, and I think it'd be really cool, and it'd be a good way for JPIDS to really get into more of a pathogenesis lane. And so you can take it from there. Tag, you're it.
Yeah, that's I do remember that. Actually, I also remember that I was coming to see an ad hoc patient with HSV encephalitis, actually, and I had brought my very first graduate student in my lab to come and see that patient that day, because I think it's really important for the PhD students to see patients who are affected by the diseases that they spend all their waking hours studying. So yeah, I obviously, this fits well with the work that I do. So we're really interested in understanding virus-host interactions between HSV, herpes simplex virus, and the brain. We're really interested in studying patients and understanding the pathogenesis of the virus, specifically within the brain, and how it gains an advantage in that environment. But I was more excited as well when we kind of broadened this to include not just herpes simplex virus, but other viruses that have a pathogenic role in the brain. And as we spoke more about it, we kind of decided to take this approach where we included not just a clinical perspective, but sort of decided to pair basic science authors or translational science authors with clinicians to sort of write each of these articles. And the reason that I got particularly excited about that, we had a couple of patients that we sort of dealt with as an entire division who were really challenging in terms of their diagnoses with CNS pathogens. And I had a couple of really insightful conversations with external experts in these specific areas, where we really just dove in and really went down a rabbit hole, trying to understand together, from a clinician's perspective, and me as someone who knows a reasonable amount about the neurovirulence of different pathogens, but then really talking with an expert who understands the complex mechanisms that are involved in these virus-host interactions or pathogen-host interactions, where they can really in this case, gave me some insight into like the real nuts and bolts of the underlying pathogenesis and how we could utilize that information in the context of what we knew about the patient to sort of make some headway in terms of this disease process that was happening with this specific patient. And that really gave me the insight that we all at some point need a little more, right? Like we need a little more understanding than we have at a basic level than we got in medical school or even our pediatrics residency or our infectious diseases fellowship. We sometimes need a little more to understand more about the patient in front of us. And so that made me want to really hear from experts all across the country. We can talk a little bit more about how we conceptualize these articles to kind of really give both clinicians a little bit more about the pathogenesis, but also to give basic researchers a little bit more about the problems that we're seeing in the clinic with these patients.
That sounds great. Anita, you want to add to that?
Yeah, I mean, you guys were the brainchild for this, right? Lisa reached out to me and said, would you like to participate? And as an ID doc and a virologist who thinks about infections in the brain on a regular basis, I thought it was a really cool topic to focus in on, really because the infection of the brain, whether it's a virus or a bacteria or a parasite, is of major clinical impact, right? The patients who have infections in the brain really have severe disease and can suffer long-term sequelae. So I think understanding this particular host pathogen interaction is super important for both the scientists and clinicians.
All right, love it. So do you two want to talk about how you sort of put the topics together, how you thought about what needed to be included?
Yeah, I think it was a little bit of kind of brainstorming together. We wanted to approach this, this is a collaborative set of articles between JPIDS, but also JID. And so we wanted to capture some pathogens that were important in the pediatric space and some pathogens that were important in the adult space, and then sort of all the nuance that's all in between, and specifically covering some pathogens that cross those barriers and maybe have different presentations in different age groups. And so, we sort of just thought about this longitudinally in terms of, from the, in utero fetus to the, person who's, quite mature. What are the pathogens that are the most important players within the CNS?
Yeah, and we
Want to
also
add to that?
really tried to focus in a lot on pathogens that are of the greatest sort of frequency of impact, right? We wanted to talk about not obscure things that rarely happen ever. So, we don't have anybody talking about Nipah, for example, right? And that clearly causes a viral encephalitis, but it's not a topic of a major article. So we focused in on things that are probably things that clinicians will see in the course of their practice.
I love it. So in as you think about people who are clinicians, sort of reading these articles, you sort of referred and we talked about the idea of wanting to pair basic science people with clinical experts. But as a sort of everyday clinician, and I don't mean that in a pejorative way, but there are some who are obviously very focused on the patients they see and really just want to kind of have the answer about how to best manage their patient. How would you suggest that they read these articles? What should their perspective be?
Yeah, I mean, I think we tried to deliberately pair, as I said, clinicians with basic or translational scientists, specifically so that we could cover topics that would be important to clinicians, and that would be written with language that was sort of tractable for clinicians. I think that in general, authors have tried to include to kind of go, like I was saying before, like one layer deeper. In general, 3,000 words doesn't give authors space or desire to go completely down the rabbit hole into areas that clinicians might not need to know anything about at all. But it does give them enough room to go one layer deeper and talk about why we use the therapies that we use, why, what is on the horizon? What do we know now that we didn't know 5 or 10 years ago about that virus host interaction that's giving us a little bit of a hook into what the next therapy might be. And so I think that clinicians should expect to look at these articles as, giving a little bit of rationale in terms of what we do now based on the underlying pathogenesis, but also what does the future hold.
Anita, you want to add anything?
Yeah, I did want to add one thing. So I think it's really important for doctors to know how to diagnose and manage CNS infections, right? That's obviously critical. But it gives you an entirely new perspective if you understand how the pathogen causes the disease than to just say, this is the disease and this is what I do about it. And I want to draw the potential readers to one of our first articles in our series, which is one by Doctors Freeman and Messakar in the January issue of JPIDS, where they talk about enteroviruses and paracoviruses. And if you're a general pediatrician, you see a lot of neonates with enteroviruses and more nowadays with paracoviruses. So this is really relevant to your practice. And in this article, they summarize and introduce AFM, acute flaccid myelitis, which is something we didn't really think a lot about until 2014, when all of a sudden there were all these cases of AFM. And so they start in this article to delve into why does this happen and what is the relevance of this? And they pull from basic science from the OG enterovirus, i.e. polio, right? This is the one that started it all off, which absolutely causes acute flaccid myelitis by definition. So they take from the basic science to teach the clinician something about the clinical picture. So I really think the readers will enjoy that article in particular.
Awesome. So thanks so much. So maybe we can just finish off with just some of your comments about as you've been receiving and curating these articles, what sort of future work do you think is still out there? I mean, I know obviously the brain is still very much a black box, but is there anything that's really struck you as you guys put this together, like these big gaping holes or undiscovered territory, or both are obviously within the field?
Yeah, I think for me, it's just the sort of entire area of, I've said this many times, but these are all of these articles are focused sort of on this pathogen immune interaction. And I think we're learning more and more, not just how to fight the pathogen, but how to fight sometimes the overactive immune response that's causing some of the pathogenesis. And I think we're learning more and more about how that immunity itself, which is supposed to be protecting the brain and is protecting the brain, can also cause pathogenesis that needs to be sort of carefully and thoughtfully and specifically narrowly managed so that your immune system is still able to fight off that pathogen, but try to decrease as much as possible that the pathogenic effect of immunity. And you need to know a lot of science in order to do that in the right way. And I think we're learning more and more about that. But for me, that's one big area where, for all of these pathogens in their own specific way, I'm excited to see how that evolves in the future.
Yeah. When you first asked us this question, I was actually thinking more along the lines of how important it is for scientists and clinicians to engage together in any research area. Quite honestly, it's not just limited to neurovirulent pathogens or even to infectious disease. I've always thought that if NIH was really smart, they would find a doctor who studies a thing and a scientist who studies a thing and intentionally pair them together, because the two together can be so much more powerful in their, directing what are the research questions we should be asking and how is this having relevance to the clinical care. But as you asked this question just now, it occurred to me that getting drugs into the brain, I think, is probably one of the biggest gaps in our field when we think specifically about infections in the brain, because we have so much trouble treating these infections because we can't get the drugs to where we want them to go, because it's really hard to cross that blood-brain barrier. Not for the pathogens, apparently, but very hard for the drugs. So I think that's an area in which I would love to see new research expanding to help us combat these CNS pathogens.
Yeah, awesome. Terrific points. All right, any last parting thoughts from either of you?
I think this has just been a really fun endeavor to try to, pair these authors and see the articles that they've produced. So I hope it's really enjoyable for, both clinicians and basic scientists to read these articles and hear the topics that these experts think are the kind of most important cutting edge topics going forward.
Yeah, it's been a lot of fun, and I'm looking forward to reading the articles I didn't get to review because I wasn't the editor for them. So we get the full spectrum now.
Well, so again, thanks to both of you for doing this. As Lisa mentioned, the article started to appear in the January issue of JPIDS, and I think throughout probably the first half of the year and maybe partly into the second half of the year, they will ping pong between JPIDS and JID, depending on the topic and how we've slotted them and when they arrive and manage through the review process. I do just also want to highlight for our listeners and readers that we are looking for new ideas. We want to have many more series like these in the future. So if you want to champion an idea of your own, if you were in your own clinic having a discussion with your colleague about, hey, this would be a great idea for a series of articles, please send us a short proposal. And we'd be happy to look at it, talk with you, and sort of flesh out the idea. And um, so again, we're really excited to see this going forward for all the reasons that both Lisa and Anita mentioned. So again, thanks to all of you. Thanks to our listeners for joining, and we'll see you next time.